aggregate persist longer, without being broken up by wave action or dissipating by diffusion. It may have helped the aggregate grow or even helped the information molecule replicate. With the presence of heredity, everything changed. Any mutation that caused the production of a more efficient, more advantageous enzyme or structural protein provided a strong selective advantage and could be passed on to progeny aggregates. Currently, attention is focusing on RNA as the first heritable information molecule. As the aggregate increased in size, perhaps largely by absorbing material from the ocean, its information molecule would replicate. If the aggregate divided into two by either wave action or surface tension, and if each half contained an information molecule, reproduction occurred. Early metabolism. The aggregates would have been complete heterotrophs, absorbing all material from the ocean and modifying only a few molecules. As aggregates continued to consume certain nutrients, however, scarcity occurred. Any aggregate that could produce an enzyme capable of synthesizing the scarce molecule from an abundant one still available in the ocean would have had a strong selective advantage. With this, there would have been a metabolic pathway 2 steps long involving 2 enzymes. Being able to synthesize a valuable, scarce molecule from an abundant, free one would have given that aggregate great advantage over the others. It would have had a more rapid metabolism and would have grown and reproduced more rapidly. Figure 17.23. We believe that many metabolic pathways evolved backward as raw materials in the environment became scarce. Currently, the pathway for photosynthesis has gone as far as it can, requiring only water and carbon dioxide. As its abundance increased, it and its descendants would finally have become so numerous as to use the second chemical faster than it could be formed chemosynthetically, and scarcity would have occurred again. Natural selection would again favor any genetic system that could extend the metabolic pathway another step to include an abundant precursor. Recall that in photosynthesis in modern plants, the only 2 precursor molecules that need to be drawn from the environment are water and carbon dioxide. Both are abundant and cheap. From them, metabolic pathways extend and ramify such that all carbon-containing compounds are derived from them. No preformed organic molecules are necessary. Energy metabolism must have been important also, and glycolysis must have evolved early because it is present in virtually all organisms. The aggregates and first cells may have absorbed some ATP and generated more by fermentation with glycolysis for millions of years. At some point, electron transport systems and hydrogen ion pumping systems would have evolved. The first would have been powered by light, and an early form of photosynthesis would have evolved. Higher plant photosynthesis is elaborate, but much simpler types occur even today in certain photosynthetic bacteria. The evolution of photosynthesis would not have been as complex or as difficult as chloroplasts would lead us to believe. Oxidative electron transport, as occurs in mitochondria, could not have evolved because free molecular oxygen still did not exist. Oxygen. The evolution of chlorophyll A and photosynthesis that liberates oxygen had 2 profound consequences. It allowed the world to rust, and it created conditions that selected for the evolution of aerobic respiration. Until that time, photosynthesis had involved the bacterial pigment bacteria chlorophyll, which liberates sulfur from hydrogen sulfide rather than oxygen from water. Earth retained its reducing atmosphere, and chemosynthesis of complex precursor molecules could continue. However, after chlorophyll evolved, the raw material for photosynthesis became water, and free molecular oxygen, O, was released as a waste product. We know this evolutionary step occurred 2.8 billion years ago because the oxygen rapidly combined with iron and formed ferric oxide, rust. Sedimentary rocks of this age contain a thick red layer of rust, indicating when oxygen liberating. Photosynthesis arrived and how long the oxygen-iron reactions continued. Only after all of the free iron in Earth's oceans had oxidized did oxygen finally begin to accumulate in the atmosphere. The atmosphere present today was derived from the early second atmosphere by this addition of oxygen from photosynthesis. It is an oxidizing atmosphere. The period of rusting was critically important for all life because it kept the concentration of free oxygen very low. It can be difficult for us humans, obligate aerobic animals, to appreciate just how toxic free oxygen is. We need it to live, but our bodies have numerous mechanisms to keep it under control and to prevent it from reacting at random in our bodies. The same is true of plants, fungi, algae, and many bacteria. Had iron not been present, the concentration of free oxygen in water and air might have increased rapidly and killed everything. Possibly there would have been no survivors. With iron, however, oxygen concentration remained low for millions of years. It was an environmental danger, but was not instantly, universally lethal. Any mutation that produced a mechanism that could detoxify oxygen had great selective advantage. Of course, the best way is to add 2 electrons to it and let it pick up 2 protons and turn into water, which is exactly what cytochrome oxidase does. This is the last electron carrier in the mitochondrial transport chain, and with its evolution, aerobic respiration became possible. Oxygen was transformed from a dangerous pollutant to a valuable resource. The evolution of aerobic respiration did not occur in mitochondria, but rather in bacteria, and some species seem to retain very relictual, early versions. The buildup of atmospheric oxygen had other important effects. Under the influence of UV light, oxygen is transformed into ozone. Ozone is extremely opaque to UV light and thus prevents most of it from penetrating deep into the atmosphere and reaching Earth's surface. This immediately removed UV light as an energy source and must have greatly slowed the chemosynthetic formation of organic molecules. Biological photosynthesis became the main means of bringing energy into the living world. The lack of UV radiation made the surface a safer place to live. UV radiation damages nucleic acids, and while the atmosphere lacked ozone, probably nothing lived near the top of the oceans or on land. With the ozone shield, organisms could move higher in the oceans and onto the mud of seashores. The transition to terrestrial life became possible. The presence of life. The chemosynthetic theory postulates a long series of slow, gradual transitions from completely inorganic compounds to living bacteria. At which stage can we say that life came into being? Can the aggregates be considered alive? This is a difficult question because the theory delineates no absolute demarcation between living and non-living objects. The chemistry of living creatures is more complex than that of non-living objects, but it does not possess any unique properties. The physics of living and non-living systems is identical. As so often in the living world, we are dealing with a continuum, not simply 2 mutually exclusive alternatives. To look for a dividing line would be simplistic. It is more important for us to understand the processes in their complexity. Figure 17.24 of the fossil remains of an early prokaryote that lived approximately 3.5 billion years ago. At the next level. 1. Isolation barriers. This chapter introduced the concept of abiological and biological reproductive barriers. The concept is simple: anything that prevents 2 members of the same species from interbreeding is a reproductive barrier. But think about this for a minute. Numerous phenomena can prevent 2 individuals from reproducing, and these phenomena might differ for long-lived plants versus annuals, for ones that live in sparse populations versus dense populations, for animal-pollinated plants versus wind-pollinated plants, and so on. There is a large literature about isolation barriers, so be careful to focus on those that pertain to plants. 2. Refugia. From time to time, conditions change such that most members of a species are killed, or at least they fail to reproduce. This might be climate change, drought, or the outbreak of a new fungal pathogen or an insect pest. During some of these changes, some small groups of individuals might be located in several safe areas called refugia, where they are protected from the altered conditions. The group of individuals may be isolated in the refugia for just a few years until the fungus disease is over, but they may be isolated as long as 100,000 years during an ice age. Either way, once the harmful conditions are over and this species can spread and survive away from the refugia, an important concept is that most of the genetic diversity of the species was lost, all except that of the members of the refugia. Consequently, as a species spreads out of refugia, it will be very different genetically than it was before the harmful conditions began. In some cases, this can lead to rapid evolution, in other cases, it results in extinction. 3. Molecular clocks and the rates of evolution. It is natural to wonder when particular organisms evolved, or when particular structures or physiologies came about. In some cases, we are lucky enough to find fossils that give us some clue, but in many cases, there are no appropriate fossils, and we have to estimate from other evidence. Many people have assumed that certain sequences of DNA, such as introns and the spacers between genes, have no function. If that is true, mutations in these regions are neither beneficial nor harmful, and these mutations do not cause a group of organisms to evolve more rapidly or slowly. The mutations merely come into existence at some neutral rate. Thus, older sequences would have more mutations than younger sequences, and by comparing related non-coding sequences in different species, we would know which species are younger and which are older. This is called a molecular clock sequence, and although some people have great faith in molecular clock sequences, other people see many serious problems. Summary. 1. When considering the reproductive effort of an entire population, it is important to consider all possible alleles for each gene, their relative frequency in the gene pool, and their effects on reproductive success. 2. Sexual reproduction alone does not cause relative abundance of alleles in the gene pool to change. Mutations bring about new alleles, but do not greatly affect allele frequency. 3. Both artificial and natural selection increase the fitness of a population to its habitat by preventing certain individuals from breeding and passing their alleles on to the next generation. Artificial selection often increases the plant's fitness for artificial habitats. 4. In order for natural selection to operate, a population must produce more offspring than can possibly survive in the environment, and offspring must be genetically diverse. These two conditions occur almost everywhere in nature at all times. 5. Natural selection operates only on the phenotypes of pre-existing alleles. 6. Plants and most other organisms never do anything with purpose or intent. Only humans and higher primates have purpose, intent, and will, and then only with regard to voluntary actions. 7. Organisms face multiple selection pressures and must be adapted to all aspects of their environment. Certain aspects are more important than others, and it may be less dangerous to be poorly adapted to one factor than to another. 8. Most adaptations have negative consequences as well as advantageous ones. Retention or loss of these alleles from the gene pool is related to the relative benefit versus cost of the adaptation. As an environment changes, the selective value of the phenotype may also change. 9. In phyletic speciation, all of a species evolves into one new species. In divergent speciation, part of a species evolves into a new species while the rest of the population continues as the original species or evolves into a different new species. 10. Gene flow is inhibited or completely prevented by reproductive barriers, either abiological aspects of the environment or biological aspects of the organisms themselves or of their pollinators and seed dispersers. 11. Life is believed to have originated on Earth by the process of chemosynthesis. This hypothesis postulates that reactions of inorganic compounds in Earth's early second atmosphere resulted in formation of organic compounds that coalesced into simple aggregates with rudimentary metabolism. After a system of heredity developed, evolution by natural selection made it possible for truly living cells to come into existence. Important terms: reducing atmosphere speciation, sympatric speciation. Review questions. 1. Evolution is the gradual conversion of one species into one or several new species. It occurs for the most part by mutations cause new or new to arise, which affect the of the individual. 2. Relative to a human lifetime, is evolution a process too slow or too fast for us to see it easily? 3. As the idea of geological evolution developed, it had an impact on biological thinking. Describe the impact. What was the importance of river deltas? 4. The discovery of evolution by natural selection happened at about the same time as other critical discoveries. Name some of these discoveries. Can you describe how they might have affected biological thinking? 5. Genes and inheritance have an impact on the lives of parents and their progeny, the F and F generations. Population genetics also deals with genetics, but in a different way. What is the definition of population genetics? How can the abundance of an allele be different in a population as compared with an individual of the population? 6. What is the gene pool of a species? Why are some alleles more common than others? 7. Many mutations can be corrected by DNA proofreading repair enzymes. What would happen to the further evolution of a species if a perfect repair mechanism evolved such that no mutation ever went uncorrected? 8. How does an accident differ from natural selection? Would an unusually severe drought with no rainfall at all for 50 years be a selective or a non-selective destructive force? 9. How does artificial selection differ from natural selection? Do you think the ancestors of lettuce had such soft, non-bitter leaves? Could today's lettuce plants survive in natural conditions? 10. One of the conditions necessary before natural selection can occur is that the population must produce more offspring than can possibly grow and survive to maturity in that habitat. Do you think this is true for most plants or most animals? Think about some tree that you see on campus or near your home. Estimate the number of seeds it makes every year and the number of years it might live. How many seeds will it produce in its lifetime? Do you think it would be possible for them all to germinate, grow, and reproduce? 11. Another condition necessary for natural selection to occur is that progeny must differ from each other in their types of alleles. Do you think this is true for most plants or animals? Be careful for some familiar crop plants. Bananas have no seeds. They must be propagated by taking cuttings from buds at the base of the plant. All of the bananas in a store are clones, they are all the same plant. Can natural selection affect these bananas? Most apple varieties are also clones. Are they being affected by natural selection? 12. Many people assume that natural selection depends on something outside the plant and the environment affecting the plant, but is that true? Wood is always inside the plant. It is never in contact with the environment. Can the phenotype of wood affect a plant's fitness? Its survival? If a mutation caused a plant to not be able to make either vessels or tracheids, just fibers, in its wood, do you think that would affect the tree's fitness compared with all of the other trees in the population? 13. Name several factors that are not part of natural selection. 14. Change each of the following sentences into ones that are true. Plants produce root hairs in order to absorb more water. Plants close their stomata at night to conserve water. DNA produces mRNA in order to keep ribosomes and potentially harmful high-energy enzymes away from itself. Stems and roots produce bark to keep pathogens out. Cacti and euphorbias have succulent bodies in order to store water. Certain plants have C metabolism in order to reduce photorespiration. Some plants time their flowering by night length in order to avoid damage caused by blooming too early in an unusually mild spring. 15. Are plants subjected to just one type of selection at a time, or are there multiple selection pressures affecting a plant? The text gives an example of a mutation that improves cold hardiness, but that might be eliminated from the gene pool despite being beneficial. Why might it be eliminated? 16. Why is it that evolutionary changes that result in the loss of a structure or metabolism can come about quickly? 17. Modern cacti evolved from ancestors that were leafy trees, and in the early stages of cactus evolution, leaves were lost. However, the capacity to produce leaves was not lost quickly. Why not? 18. Look at the two cacti in Figure 17:11. Both the leafy Periskia in Part A and the very desert-adapted Gymnocalycium in Part B evolved from the same ancestors, leafy trees that were not themselves cacti. In which genus, Periskia or Gymnocalycium, has natural selection favored more of the new alleles produced by mutations? In which genus has natural selection retained more of the original alleles and eliminated almost all of the new alleles produced by mutation? 19. What is phyletic speciation? What is divergent speciation? 20. What is gene flow? How does it occur? 21. The Hawaiian Islands were formed as underwater volcanoes that became so tall that they protruded above the ocean surface. Being formed from molten rock, they were absolutely sterile at first, but now they are home to many types of plants and animals. How did the ancestral plants and animals get to Hawaii? Was this a type of gene flow? 22. What is an abiological reproductive barrier? Name several. Is something that is an abiological reproductive barrier for a plant necessarily also one for an animal? Would you guess that small herbs have more abiological reproductive barriers than do birds? 23. Differences in flower color, shape, or fragrance are listed as being potential biological reproductive barriers. Do you think they would matter at all in a wind-pollinated species? If a plant species has several flower colors, could a mutation in the plant's pollinator, but not in the plant species itself, be a biological reproductive barrier? For example, a mutation that makes it unable to see red? 24. One of the earliest postzygotic barriers to arise is hybrid sterility. Two plants can be crossed, but the resulting seeds grow into sterile plants that cannot reproduce. Is this as important in plants as it is in animals? 25. What is genetic drift? What is a founder? Are these important in large populations or in small ones? How are these phenomena important when we create national parks and preserves to protect endangered species? 26. What is convergent evolution? Euphorbias and cacti are often used as examples of convergent evolution. Have their genotypes converged? Have their phenotypes converged? Both have spines, but if we look carefully we can tell that those of cacti evolved from and those of euphorbias evolved from. They have converged, but not so much that we cannot tell them apart. 27. What is the theory of chemosynthesis? 28. Describe the conditions present on Earth at the time of its early second atmosphere. What chemicals and energy sources were present? What chemical in particular was absent? Can you speculate about what conditions would be like on Earth today if meiosis, crossing over, and sexual reproduction had never evolved? 29. Life originated on Earth so long ago that erosion and weathering and metamorphosis of rock have destroyed most traces of the processes that occurred, but oxygen is an interesting exception. What is the source of all the oxygen in our atmosphere? What happened to the first oxygen to be produced? Why was this important to the survival of all other life forms? Design credits: Hummingbird, Tango 58 Moment Getty, Green Plant Cells, Shutterstock Incorporated Natalia Ora, Purple Tulip, Shutterstock Incorporated Maurice C. Fields, Dandelion, Shutterstock Incorporated Daniel Kreisel, Poppy, Shutterstock Incorporated Saryuri, Plant Icon, Shutterstock Incorporated Vector, Digging Man Icon, Shutterstock Incorporated ZArt. Chapter 18. Classification and Systematics. Chapter opener image. You probably have no trouble recognizing these as palm trees, even though it is unlikely you have ever seen these plants before. They are Washingtonia filifera, native to seeps and artesian pools in the deserts of California. They are recognizable as palms because they have so many features we associate with palms we are already familiar with: a solitary unbranched trunk topped by many large leaves. Most palms have these features because they arose early in the evolution of the palm family and have been inherited for generation after generation ever since. But habitats are variable and plants diversify, so some palms are now shorter, others grow in clumps, some have smaller leaves. Features such as having one unbranched stem have evolved in other families. For example, many people assume that cycads are short, fat palms. When we attempt to classify organisms, we try to group together those that have similar features because they are closely related, and we try not to be fooled by characters that have evolved 2 or 3 times in different groups. Outline. Concepts. Levels of taxonomic categories. Cladistics. Understanding cladograms. Cladograms and taxonomic categories. Other types of classification systems. Artificial systems of classification. Classification systems for fossils. Taxonomic studies. The major lines of evolution. Box 18-1 Plants and People, Development of Concepts of Evolution and Classification. Box 18-2 Botany and Beyond, Cacti as examples of evolutionary diversification. Box 18-3 Plants and People, Genealogy versus Clades, Your family history is the opposite of a clade. Box 18-4 Plants and People, Identifying unknown plants. Learning objectives. After reading this chapter, students will be able to order the levels of the natural classification systems, explain synapomorphies and homoplacies, summarize the way a cladogram shows evolutionary patterns, define paraphyletic groups and polyphyletic groups, recall 2 uses of an artificial classification system, summarize the process for declaring a new plant species, restate the importance of isotypes, name the 3 domains of all organisms, list the major evolutionary lines that diverged within kingdom Plantae. Did you know? All plants evolved from one ancestral set of plants, so all are related to each other and have one common ancestor. Until the 1800s, anyone could make up their own name for a plant or animal. Names were very confusing. In the mid-1800s, various scientific societies decided they would accept only one set of names, those based on the work of Carolus Linnaeus. Ancient manuscripts in the cuneiform of Sumerians and hieroglyphics of Egyptians name numerous plants, but without pictures, we do not know which plants these refer to. Concepts. Starting approximately 400 million years ago, a long series of mutations in natural selection gave rise to the plants that are alive today. During the intervening eons since then, that evolutionary line has progressed and diversified, branching into more and more lines of evolution. Thousands of these have become extinct and are known only by fossils. Thousands of other evolutionary lines are represented by the approximately 280,000 species of living plants. The hereditary relationships of any group of organisms constitute its phylogeny, basically the evolutionary history of each member of the group. Some phylogenetic studies attempt to create models of the evolutionary relationships of very large groups. For example, the steps that were involved as plants, animals, and fungi evolved from some early ancestor. Other studies focus on the relationships of smaller groups, such as the species of the legume family. The goal of modern plant systematics is to understand each of these evolutionary lines and to have a system of names, nomenclature, that reflects their relationships accurately. In the past, plants were given names merely so that people could communicate about medicinal plants, crops, poisonous plants, and so on. The task of taxonomists was to discover and identify new species and give them unique names, but with the discovery of evolution by natural selection, the basis of naming plants suddenly changed. Natural selection showed that all organisms are related to each other genetically, all are part of a large phylogeny. Taxonomists, scientists who specialize in classification and naming, immediately realized that the most scientifically valid system of assigning names to species would be one that reflected evolutionary relationships. At the end of the 19th century, taxonomists adopted the goals of developing a natural system of classification, a system in which closely related organisms are classified together, and assigning plant names on the basis of phylogenetic relationships. The nomenclature would reflect the natural system of classification. Because organisms range from closely to distantly related, taxonomists devised a classification and a nomenclature with numerous levels. In this system, closely related species are placed into a genus, and closely related genera are grouped together into a family, and so on. Today, as in the past, there are many regions of Earth that have not been adequately explored. These probably contain thousands of plant species that have never been collected, named, or studied. Plant systematists still have the goals of searching for new species and using them to construct more accurate models of plant evolution. It is important to remember that the pattern of evolutionary diversification, the phylogeny, is a reality. Our classification systems are hypotheses, models that attempt to map that evolution. Because our knowledge is incomplete and imperfect, the current classification systems are only approximations. Most are probably close approximations, and we do not expect major changes. However, we do expect numerous smaller changes to be made periodically for many years. Levels of taxonomic categories. Plants have varying degrees of relatedness, and a natural classification system reflects this in its numerous levels. The most fundamental level of classification is the species, which ideally and theoretically is a set of individuals closely related by descent from a common ancestor. Members of a species can interbreed with each other successfully but cannot interbreed with individuals of any other species. Some species are not so predictable, they may not interbreed well with closely related species, but an occasional cross-pollination results in a viable seed that grows into a fertile adult. If this occurs frequently, the two plant groups may best be considered subspecies of a single species. The two subspecies must be so similar genetically that the chromosomes of the two parents can function in the same nucleus, and when the plant is mature, spore mother cells can undergo meiosis successfully. For all of this to happen, the organisms must be extremely closely related, having undergone divergent evolution from a common ancestor only recently so that few mutations have accumulated since they diverged. But as mutations continue to accumulate in each subspecies, finally either no viable seed results when the two are crossed or the seed grows into a sterile plant. The two then must be considered separate species. Closely related species are grouped together into genera. Deciding whether several species are closely related enough to be placed together in the same genus is difficult. No objective criteria exist, the decision is entirely subjective and is often the cause of great dispute. Some taxonomists, generally referred to as lumpers, believe that even relatively distantly related species should be grouped together in large genera. Other taxonomists, called splitters, prefer to have many small genera, each containing only a few species that are extremely closely related. For example, some taxonomists believe that cranberries and blueberries are so similar that they should go into the same genus, Vaccinium. Others think that cranberries are distinct enough that segregating them into their own genus, Oxycoccus, more accurately reflects evolutionary reality. Both groups of taxonomists agree that the two sets of species are closely related, but they have different opinions as to how much evolution has occurred since the time of the most recent common ancestor. The critical concern is that the genera are natural, that all of the species included in the genus are related to each other by a common ancestor, and that all descendants of that common ancestor are in the same genus. Such a group is monophyletic. In an unnatural, polyphyletic group, members have evolved from different ancestors and may resemble each other only as a result of convergent evolution. Whenever systematists discover that they have accidentally classified unrelated plants together in an unnatural, polyphyletic group, they immediately search for a more accurate classification composed only of monophyletic groups. Table 18:1 The taxonomic categories. Plants and people. Box 18:1 Development of concepts of evolution and classification. Our modern classifications have their origins in ancient Greece. Theophrastus, a student of both Plato and Aristotle, wrote extensively about plants around 300 BC and established many important concepts. He distinguished flowering and non-flowering plants, recognized sexuality in plants, and understood that fruits develop from carpels. Theophrastus described almost 500 species, and our genus names Asparagus, Narcissus, and Daucus can be traced directly back to him. Pliny the Elder, a Roman lawyer and natural historian, wrote voluminously on almost every subject. His largest work, Natural History, was an attempt to describe everything in the world. Despite its inclusion of many fanciful creatures based only on folk tales, it served as the definitive, authoritative source of information on most subjects for more than 1,000 years. The most important book on plant classification from the ancient world is Materia Medica by Dioscorides. Written in the 1st century CE, it describes 600 plant species and how they can be used to treat disease. It was the best, often the only, source of information about preparing herbal medicines. Materia Medica was at last superseded by more accurate work during the Renaissance, but it is still published today for its immense historical value as a direct link to early Greek science. Between the time of Dioscorides and the Renaissance, almost no works of great value in natural history were written. Changes began to occur in the 15th century, and Europe entered an age of exploration. Prince Henry of Portugal sent ships on expeditions down the west coast of Africa, and the explorers returned with new plants and animals and knowledge of new lands and peoples. Simultaneously, exploration of physics, chemistry, astronomy, and geology began. An important result of these explorations was the discovery that Pliny and other ancient authorities had been wrong in many subjects. Until then, people thought the ancient Greeks and Romans had represented a golden age and were basically infallible. The realization that the ancients had made errors meant that the answers to questions about the world had to be sought in the world itself, not in ancient books. Exploration, not only geographical but also scientific, philosophical, and religious exploration, became an obsession. In botany, this new, independent thinking first became apparent in the publication of medically oriented plant books, called herbals. By the middle of the 16th century, they began to contain careful, precise descriptions of plants based on first-hand observation of plants, not by paraphrasing Materia Medica. Most herbals contained fewer than 1,000 plants, so a reasonably good botanist could become familiar with all of them. However, as exploration continued, especially after the discovery of the Americas, the number of plant species became too large for this type of familiar treatment. It became necessary to develop a classification system so that a person would have some means of identifying an unfamiliar specimen and finding it in the ever larger herbals. Several important ideas developed at this time. One was the concept of a genus as a group of similar species, established by Gaspard Bauhin. Although this may seem like an obvious notion, it was a profound breakthrough at the time. The concept of species had been easy. God was believed to have created all of the types of organisms, the species. But this concept did not explain why there should be groupings of species, as the concept of genus implies. Why would God create several types of roses, several types of mints? This was not a trivial question then. Until the theory of evolution by natural selection, the world was viewed as a reflection of the mind of God, who presumably had created it. If there were thousands of species of grasses, but only 3 of cattails, this would reveal something about how God thinks. It was obvious to anyone familiar with the increasing number of plant and animal species that some species resembled each other very closely. But why? Our system of nomenclature, of scientific names, can be traced directly to Carolus Linnaeus, a professor of natural history at the University of Uppsala in Sweden during the middle and latter part of the 18th century. He adopted the genus system of Bauhin, created a large numbers of genera, and placed every species into one genus or another. Every species had both a genus name and a species epithet, the basis of our present binomial system of nomenclature. Before Linnaeus, rather than names, plants had paragraph-long descriptions of their features, which was obviously cumbersome. After Linnaeus, each plant had a name of just 2 words. Figure B18, 1a. Prior to the Renaissance, most European botanists considered ancient books to be a better source of knowledge than plants themselves, so they merely copied drawings from pre-existing books, and each drawing became less accurate and less useful. Left, cardamom; right, capparis; bottom, cassia lignea. In 1753, Linnaeus published Species Plantarum, a treatment of all plant species known in the world at that time. This book made botanical studies of all types much easier because specimens could be identified quickly. However, the system often placed unrelated species into the same genus on the basis of homoplasic characters. The idea of the evolution of species developed slowly over many years. It did not begin with Darwin and Wallace by any means. Bauhin recognized resemblance early, and an expansion of the idea of similarity occurred at the Jardin des Plantes in Paris. Antoine Morin de Jussieu wanted to organize the garden in a logical way so that plants resembling each other were planted together. The accompanying catalogue was Genera Plantarum. It was the first major attempt at a natural system of classification, and people could not help but wonder whether some species within each genus had not evolved from others. Perhaps similar genera had changed, evolved. But how? Science was still dominated by the idea of divinely created types. Unfortunately, J.B.P. de Lamarck at this time presented his theory of evolution by inheritance of acquired characteristics. This was the incorrect idea that all cells of the body produced fluids that diffused to the genitalia, where the fluids were concentrated and formed into sperm cells or egg cells. As an individual changed, the secreted fluids and therefore the characteristics carried by the gametes would change. The evolution of giraffes was explained as follows. Ancestors to giraffes had been born with short necks. However, as they stretched their necks to reach leaves in high trees, their necks lengthened. Long necks in the adults would be the acquired characteristic, and subsequently, the genetic fluid passed to their gametes carried information about long necks. Consequently, their offspring had longer necks. Mendel showed that this could not be right because genes are non-varying particles, not variable fluids. Evolution by natural selection explains giraffes by postulating that variability of neck size occurred among the earliest ancestors of giraffes. Alleles in some animals produced short necks and in others slightly longer necks. Those with longer necks could reach more leaves, therefore, they had better survival and reproductive success. Over millions of years, various mutations produced new alleles that resulted in even longer necks, and these new alleles were increased by natural selection. The theory of inheritance of acquired characteristics was never widely accepted, and it gave evolution a bad reputation for many years. Figure B18, 1b. During and after the Renaissance, people realized they could make accurate drawings and studies of plants only if they examined the plants themselves. Much more realistic images were produced. These are drawings of tobacco plants. In 1859, Charles Darwin and Alfred Wallace each propounded the theory of evolution by natural selection, which gave natural systems of classification an immediate validity. Taxonomists quickly understood why some species within a genus resemble each other so strongly. They are descendants of a common ancestor and are related by evolution. Similarly, closely related genera constitute a family. The theory of evolution by natural selection became accepted while two German botanists, A. Engler and K. Prantl, were working on a monumental classification of all the world's plants, Die Natürlichen Pflanzenfamilien, published in 1915. It organized species on a phylogenetic, natural basis, as understood by Engler and Prantl. In those early days of evolutionary studies, organisms were assumed to have evolved from the simple to the complex. Thus, in flowering plants, the wind-pollinated species that have no sepals and petals were placed first. The sunflowers, with their complex floral structures, were considered more advanced. It was quickly discovered that simplicity may also be the result of evolutionary simplification. Because no automatic correlation exists between simple and primitive or between complex and advanced, the Engler and Prantl classification has many errors, although as a compendium of information it is still an unmatched vital resource. Other major classifications were produced by C. E. Bessey of the University of Nebraska, A. Takhtajan in Russia, and A. Cronquist at the New York Botanical Garden. The level above genus is family, each family being composed of one, several, or often many genera. Most families are well defined, with widespread agreement as to which species and genera belong in a particular family. As examples, consider how easy it is to recognize the following families: cacti, orchids, daisies, palms, and grasses. The reason for this probably is the age of most families. A line of evolution must be very old, usually at least several tens of millions of years, to diversify into several genera and many species. Even closely related families have been separate evolutionary lines for at least 20 or 30 million and often 50 million years. The common ancestor and many of the earliest species probably have become extinct. Therefore, although two families have many characters in common, enough features are unique to each family to make them recognizable as distinct from each other. The levels above family are order, class, division, and kingdom. Although you might expect universal agreement at the level of kingdom, even the boundaries of the plant kingdom, kingdom plantae, are disputed. Some believe that green algae should be included because they are almost identical biochemically to vascular plants. Others believe that, despite the biochemical similarity, green algae should be excluded because they are so different morphologically and anatomically. Except for kingdom, genus, and species, the names must have a certain ending to indicate the classification level. Division names end in phyta, for example, Chlorophyta, Coniferophyta, and Magnoliophyta. Class names end in Opsida, order names in Alesh, and family names in AC. Genera and species names do not have standard endings. The scientific name of a species is its genus and species designations used together and either underlined or italicized. For example, tomato is Lycopersicum esculentum. Note that the species name is not esculentum but is Lycopersicum esculentum. Esculentum is the species epithet, the word that distinguishes this species only from the other species of the genus Lycopersicum. We cannot refer to tomatoes as esculentum because that species epithet is used for many different species in different genera, for example, buckwheat, Phagopyrum esculentum, and taro, Colocasia esculenta. Some families have two names. The above rules were adopted to regularize family names so that each family is named after one of its genera, using the AC suffix. The rose family was named Rosaceae, based on the genus Rosa. This worked well for most families, but for some, the old name was so well known and so familiar that it was kept as well. Examples are Asteraceae, Fabaceae, Ericaceae, Poaceae, Brassicaceae, and Apiaceae. The word taxon is used to refer to any of the above groups in a general way. For example, some systematists study smaller taxa such as species and genera, others are concerned with higher taxa such as orders and divisions. Cladistics. The study of phylogeny centers on examining the similarity of one species to others. A species evolves into two species as different populations accumulate distinct alleles. Even when enough divergence has occurred to create separate species, the two still resemble each other strongly. As they continue to evolve, each acquires its own mutations, and because they cannot interbreed, they cannot share the new alleles. Thus, they differ from each other more as time passes. Distantly related plants have been on separate lines of evolution for millions of years, time enough for so many mutations to accumulate that they resemble each other only slightly. Cladistics is a method of analyzing these phylogenetic, evolutionary relationships. Cladistic studies are complicated by the fact that plants can resemble each other for two distinct reasons: they have descended from a common ancestor, or they have undergone convergent evolution. Features similar to each other because they have descended from a common ancestral feature are synapomorphies. For instance, almost all members of the Anthurium family are easily recognizable because of their spada and spodix inflorescence. All members have these structures because all have inherited their inflorescence genes from a common ancestor. The spada and spodix are homologous in all Anthurium species. Homologous features are the ones critically important for making taxonomic comparisons and the only ones that can be used to conclude that species are related. The second cause of resemblance, convergent evolution, results when two distinct evolutionary lines of plants respond to similar environments and selection pressures. Under these conditions, natural selection may favor mutations in each line that result in similar phenotypes. Features like this are homoplaces and should never be used to conclude that plants are closely related. A striking example is the convergent evolution of cacti and the succulent euphorbias. Both occur in deserts where a succulent water-storing body is advantageous, and spines are selectively advantageous by deterring animals from eating the plants to get the water. The two families are not considered closely related on the basis of spines and succulents, however, because these features do not share a common ancestry, they converged evolutionarily because of similar selection pressure. Determining whether a similarity is due to homology or analogy can be extremely difficult. In the case of spines, it is easy. Cactus spines are modified leaves that are always smooth and never branched, occurring in clusters on an extremely short shoot. Euphorbia spines are modified shoots that branch and have small, scale-like leaves. Being modified shoots, they occur singly in the axil of a leaf, never in clusters. If we look beyond the analogous similarities, the dissimilar features are dramatic. Flowers of cacti are large and have many parts, whereas those of euphorbias are small and unisexual, either staminate or carpellate. The wood of cacti is parenchymatous and has few fibers, whereas that of euphorbias is fibrous and hard. Most important, the plants that most closely resemble the succulent euphorbias in important homologous features are the non-succulent euphorbias of Africa. Succulent cacti resemble a completely different set of plants, non-succulent cacti of the tropical regions of the Americas. Thus, the evolution of spines and succulents occurred independently in each group. A corollary to the assumption that similar plants are closely related is the assumption that dissimilar plants must not be closely related. Studying lack of similarity can also be difficult because in some cases a small genetic change results in dramatic phenotypic changes. Mutations that affect production, distribution, and sensitivity to hormones result in large changes of the phenotype between two closely related species. Also, mutations that affect early stages of development such as the embryo or bud meristems can cause closely related species to look deceptively dissimilar. In many evolutionary lines, species of large woody trees have evolved into herbs. The main transition involved is the ability to flower early and then not survive a harsh winter. It must be a simple evolutionary process involving few genes because it has occurred many times. For example, nearly half the species of the Violaceae are herbs in the genus Viola, violets and pansies, but the early members of the family were woody trees, as are the members of the relictual, tropical genus Renoria. In the cabbage family, artificial selection by humans has resulted in one species that has dramatically different forms. Broccoli, Brussels sprouts, cabbage, and cauliflower are all subspecies of a single species, Brassica oleracea. For taxonomic studies, a mosaic of evidence is available, some valid and useful, some misleading. Systematists attempt to look at as many features as possible on the assumption that misleading evidence will be outweighed by valid characters. Also, certain features are considered more significant. Those that result from numerous metabolic interactions and the influence of many genes tend to change more slowly than those with a simple metabolism controlled by just one or two genes. Loss of features, such as loss of sepals and petals in wind-pollinated flowers, is often rapid because deleterious mutations occur commonly. Without the natural selection of insect pollination to eliminate these mutations, they are retained and cause a loss of perianth. After those genes have become so mutated that a flower lacks sepals and petals, the evolution of new ones would be slow and difficult. Currently, taxonomists study virtually every aspect of plants using a wide variety of tools. Simple observation of major parts is still important, but scanning electron microscopy to study hairs, stomata, cuticle, and waxes is also used. Internal structure is studied with both light and transmission electron microscopy. The nature of plastids in the phloem has been found to be an extremely valuable character. Various aspects of metabolism are important, ranging from the types of pigments in flowers to the presence of crassylation acid metabolism and C metabolism and specialized defensive, antipredator compounds. DNA sequencing is a new tool for analyzing evolutionary relationships. Two plants are considered separate species only if they differ in significant, heritable ways. Therefore, the sequence of nucleotides in the DNA of each species must differ from that of all other species. When plant phenotypes are studied, the actual objective is to determine differences in genotype. In the past, this could be done only by examining phenotypic features. Now DNA can be examined directly and mutations can be identified, even if they do not cause any detectable change in phenotype. Figure 18. Members of the family Araceae all have a spatha and spadix, as in this Amorphophallus and Anthurium. In all extant species, the genes that control the formation of these structures are descendants of the genes of the original, ancestral species. The genes that exist today have resulted from thousands of rounds of DNA replication during plant growth, crossing over, syngamy, and then more rounds of DNA replication as the zygote grows. This has been repeated millions of times since spaths and spadices first evolved millions of years ago. During all this, mutations in natural selection have led to obvious varieties of spaths and spadices, but the basic relatedness, the homology, is still obvious. Figure 18, two cactus spines are modified bud scales, which makes them modified leaves. They occur in clusters as all bud scales do, and they never branch. Disco cactus. Euphorbia spines are modified branches. They bear small leaves that may themselves have axillary buds, therefore, the spine can branch, as in the Gemmelliformis. Although the spines in cacti and euphorbia are often called homoplaces, they are so different it is difficult to confuse the two. In contrast to homoplacy, another problem arises when a structure evolves so much as to be almost unrecognizable. It took a great deal of study to be certain that cactus spines are modified leaves, and they continue to evolve. These flat, thin structures are spines of Tephrocarpus papyracanthus. They protect the cactus by disguising it. This species grows among grasses and is almost impossible to find unless it is flowering. The spines of this Tephrocarpus stabiliformis have evolved to be glands, extrafloral nectaries. Each droplet is a secretion of dilute sugar water that attracts ants to the plant. The ants protect the plant as they defend their source of food, and were cultivated by bee barth. Figure 18, 3. This small, leafless shrub may not at first glance appear to have much in common with this larger, leafy tree, but careful examination reveals homologous similarities in many critical features of the flowers and stems, which allow assignment to the same genus, Acacia. In this case, genes controlling flower and stem morphogenesis have changed little during evolution, whereas genes controlling elongation growth of stems, formation of leaf primordia, and formation of bark have all changed dramatically. Like traditional taxonomic data, DNA sequencing data have their own ambiguities and limitations. Any two species, even those closely related, must differ at many points of their DNA. Simply counting the number of sequence differences is not enough, because even individuals of a very uniform, homogeneous species may have hidden mutations that do not affect the phenotype. Furthermore, any two species in the same family probably share the great majority of their DNA sequences in identical form, with only a few genes mutated. How many differences are needed to conclude that two genomes represent two distinct species? There is no simple answer. DNA sequencing permits a new type of study, the evolution of DNA itself. Sequencing allows us to investigate the evolution not only of taxa but of centromeres, telomeres, DNA replication start sites, enhancer elements, boundaries between introns and exons, and so forth. Understanding cladograms. A cladogram is a diagram that shows evolutionary patterns by means of a series of branches. Each point at which a cladogram branches, called a node, represents the divergence of one taxon into two, and all of the branches that extend from any particular point represent the descendants of the original group. That ancestor is their common ancestor. Any ancestor and all of the branches that lead from it constitute a clade. Because each node represents a taxon dividing into two, it also represents some detectable change that creates a new group. After the divergence, one of the taxa differs from the other. For example, a plant in a species with red flowers might undergo a mutation that gives it white flowers. As long as the two can still interbreed, this is not especially significant. However, the change in flower color may cause different pollinators to visit it, and it might become reproductively isolated from its red-flowered ancestors. If the white-flowered individuals thrive and become numerous, but cannot interbreed with the red-flowered ones, this is a new species, and the divergence should be represented as a node with two branches coming from it. In this case, white flowers are a derived condition, an apomorphy, and red flowers are the ancestral condition. Table 18, two DNA sequences used for phylogenetic studies. Figure 18, for a simple cladogram showing the phylogenetic relationships between an ancestral group and its two descendants. The ancestral condition was to have red flowers. The group diverged into two. Now one group has white flowers, and the other still has red flowers. Be careful that this is treating only a very limited amount of information. It does not indicate whether we are dealing with two species and their ancestral species or whether we are comparing two genera or two families. Both populations might continue to evolve and diverge, giving rise to a phylogeny like that shown in Figure 18.5. Node 1 here represents the same taxon as Node 1 in Figure 18.4, but now with more time, both its lineages have diverged. At Node 5, some that had continued to have red flowers have diverged into two groups, one continuing to have red flowers and the other mutating to have white flowers. White flowers have now originated twice. Synthesis of pigments requires many steps and many enzymes, and mutations in any of those genes could cause the same phenotype. The two sets of white flowers almost certainly have different genotypes. This is homoplasy. The original white-flowered lineage has diverged twice. After Node 2, one group has trichomes on its leaves, and the other continues to have smooth leaves. At Node 3, dwarfism originates in one line. At Node 4, one group mutates to have narrow petals. We can speak of many clades here. All of the species depicted, plus their common ancestor at Node 1, form a clade. Species E and F plus Node 5 are a smaller clade, as are the four species that have descended from Node 2. It is especially important to notice what is not a clade. There are now 5 species with white flowers, but we cannot classify E with the others. They do not form a clade. The most recent common ancestor of all white-flowered species is Node 1, and a natural, monophyletic clade includes the ancestor and all of its descendants. If we would try to classify A, E as a group but leave out F, we would be creating a paraphyletic group, one that does not contain all the descendants of the most recent common ancestor. Figure 18.5, a more complex cladogram with more taxa and characters. We have data for the following characters: flowers, seeds, leaves, plant height, and petals. In all taxa, the fruits are berries. Imagine that the ancestral group had red flowers, large seeds, leaves without trichomes, tall shoots, and broad petals. See the text for details. Think about what a systematist is faced with. There are 6 species, some with white flowers, some with trichomes on their leaves, some with a dwarf stature, and some with narrow petals. In constructing a cladogram, we search for shared derived characters, synapomorphies. Species C indeed both have trichomes on their leaves, but no other species does. Therefore, this is a synapomorphy that shows these two are closely related. Clades A, D share the synapomorphy of having small seeds. Species A is the only one that is dwarf. No other species has this feature, so it does not help us understand the phylogeny. It is uninformative. All species still produce fruits that are berries. This has not changed anywhere. This shared ancestral condition is a simplesiomorphy, and it too is uninformative for arranging these species. Given these 6 species and these characters, a systematist could create any number of cladograms. Between Node 1 and species A, we could imagine many steps in which flower color changed from white to blue to yellow and then back to white, with the blue and yellow forms having gone extinct. This might have happened. We can see the 6 living species and their characters. However, we cannot see any of the ancestors, and there may have been other descendants that died out and thus are missing from our cladogram. Given the information we do have, we use a principle called parsimony. We prefer the simplest possible hypothesis, and we do not make a hypothesis any more complicated than it needs to be. Parsimony is used in all fields of biology. The most widely accepted theory of water movement in vascular plants is that water moves upward through xylem. We could imagine all sorts of much more complicated mechanisms, but we focus on the simplest one and move on to a more complex hypothesis only if data indicate that the simple hypothesis is not accurate. Botany and beyond. Box 18-2 cacti as examples of evolutionary diversification. The cactus family is a large group that has undergone extensive evolutionary diversification with the modification of many characters. Many of the changes are easy to see and understand, so this is a good group to use as an example. First, several morphological and DNA features unite the entire family. The most obvious is that all have spines that evolved from modified bud scales. They are present in almost all members. And no other family produces spines this way. The first divergence at node 1 produced one clade labeled Pereschia and another clade that leads to all the other cacti. The Pereschia clade actually branches into several species, all of which lack many derived characters. These are large trees with ordinary broad, thin photosynthetic leaves, hard woody trunks, and no succulent tissue. These features are what we would expect in an ordinary Eudicot tree, therefore, we think that many of these are ancestral characters, not derived ones. Pereschias are so similar to each other and so different from other cacti that they are grouped together as a subfamily, Pereschioideae. Nodes 2 and 3 lead to Meiwenia and Opuntia. Plants in these clades are very similar to Pereschias, but each has several derived characters. Plants in both are small, woody shrubs with small, cylindrical leaves that appear to consist of just a midrib without any lamina. Their cortex is thicker, with more water storage parenchyma than that of Pereschias. Opuntias differ from Meiwenias and all other cacti in having hundreds of tiny bristle-like spines that are a synapomorphy, distinguishing all Opuntias from all other cacti. Meiwenias are distinguished from Pereschia and the Opuntias by DNA sequence differences. Meiwenias are placed in their own subfamily Mehuenioideae, and the Opuntias are in subfamily Opuntioideae. Node 4 is a real surprise. One branch leads to Blossfeldia, a monotypic genus of plants that have many derived characters. They are tiny dwarfs only about 1 centimeter tall when mature. Their foliage leaves are reduced to just leaf primordia. They have stomata in their stem epidermis, and their wood has an unusual type of tracheid but no xylary fibers. Until recently, B. Lilliputana was placed up by node 10 among several other genera that have similar, highly derived features, but DNA sequencing by several people indicates that it is a sister to all of the cacti from node 5. Figure B18, 2b Pereschia saccharosa, spines are modified bud scales. Notice the large green foliage leaf. Figure B18, 2c Meiwenia poapigii. This is a cushion plant, a short, extremely compact tree approximately 20 centimeters tall. This is a common adaptation to cold, windy alpine habitats. Yellow structures are fruits. Figure B18, 2a plant of Pereschia saccharosa. Cacti from node 5 are extremely variable, but all are more cactus-like than Pereschias and Meiwenias. These really look like cacti. They are succulent, have water-conserving and water-storing adaptations. Those at node 8, Leptocereus, Pachycereus, and their relatives, still have many ancestral features, especially their hard, fibrous wood that has few adaptations for desert life. Taxa from node 6 are extremely derived, with fewer ancestral characters. They tend to have spherical or short cylindrical bodies. Many have specialized spines that shade the body, disguise it, or secrete nectar. Also, this clade occurs only in North America, not South America, and geographic distribution is often an important character. Figure B18, 2f Blossfeldia lilliputana. This is a mature, full-grown plant, approximately 1 centimeter tall. Figure B18, 2g Mammillaria lasiacantha, and many of its relatives are small, desert-adapted plants with many derived features that occur only in cacti. Figure B18, 2h Echinocactus horizonthalonius is one of many species of barrel cacti in North America. Figure B18, 2d tiny but obvious foliage leaves of Meiwenia poapigii. Figure B18, 2e Ostrocylindra puncha Schafferi has leaves without a lamina. Its wood is hard and fibrous, and it has little succulent tissue. All clades from node 9 are predominantly South American. Only a few individual species have spread to the Caribbean, Florida, Central America, or Mexico. Hashiora and its relatives are epiphytic, living only in rainforests, not a habitat we typically associate with cacti. Of the remaining clades, Parodia, Gymnocalycium, and their relatives are small, globular South American cacti that greatly resemble the North American cacti of node 6. In fact, early taxonomists often classified them together.
One of the original reasons for considering them distinct is that the two clades occur so far from each other, separated by thousands of miles of ocean and Amazon rainforest. Later, certain flower, fruit, and seed differences were noticed, And more recently, DNA sequencing also supports the idea that North American small cacti and South American small cacti are remarkable cases of convergent evolution within the same family. The most recent common ancestor of node 6 and node 10 is node 5, and plants at that node must have lacked many derived features while having many relictal ones. This family has tremendous diversity. It contains trees, shrubs, and dwarfs, succulents and non-succulents, plants adapted to deserts, rainforests, grasslands, and alpine regions, flowers pollinated by insects, bats, and hummingbirds. The monophily of the family is not in doubt, however. All are universally accepted as being members of the same family-level clade. Figure B18-2J Pachycereus pringlei has a very thick cortex and is capable of storing large amounts of water. Its wood, however, is like that of Pereschia, fibrous and strong enough to support tremendous weight. Figure B18-2I Leptocereus weingartianus is a shrubby, brushy cactus sprawling out of the undergrowth. Its stems are narrow, with little succulent tissue, and it is highly branched, like an ordinary non-cactus bush. The tall, upright cacti are Pilosocereus polygonus. Figure B18-2 Cahacior solicornioides is an epiphytic cactus with tiny, narrow stems. It never becomes a large plant. Figure B18-2 L. Parodia mossi are small globular South American cacti whose body size and shape are similar to those of Mammillaria because of convergent evolution. Figure B18-2 M. Gymnocalycium horsti has obviously diverged greatly from its Pereschia-like ancestors. Figure B18-2 Anoriocereus celsianus are adapted to high, cold alpine conditions on the border between Bolivia and Argentina. Figure B18-2 O simplified cladogram of several clades and cactaceae. In constructing cladograms, a great deal more data are used than those shown in Figure 18-5 for living species. We have DNA sequences in which each base pair is a data point, and thus, we might have dozens or hundreds of informative sequence differences when comparing 6 species. However, when a computer program uses the data to construct a cladogram, it almost always comes up with several that are equally simple, equally parsimonious, but that have the taxa arranged differently. When this happens, the various cladograms are analyzed to see if any clade appears repeatedly. If so, we have confidence that that particular clade might be an accurate depiction of the phylogeny. Sometimes there is simply not enough data, and then usually the various species are shown as arising from the same node. This is an unresolved polychotomy. These become the focus of more intense study. As you study cladograms, keep in mind that they are hypotheses. Each is a model supported by some evidence, but we do not have all possible information about all species either living or extinct. DNA sequencing projects are progressing very rapidly and providing large amounts of data. These can be combined with morphological, anatomical, and physiological data to refine the cladogram and to understand not only how taxa are interrelated but also how the characters themselves have evolved. Table 18-3 Cladistics. Cladograms and taxonomic categories. How are names or taxonomic levels assigned to a cladogram? The only taxonomic unit with an objective definition is species, individuals that can interbreed. But there is no such objective definition for genus or family or so on. It has been proposed that every node and every clade in cladograms be names, but that would result in an unacceptably large number of names. Even worse, most cladograms are still not stable, they change as new species and data are added. Many systematists have decided to continue using the old names for genera, families, and orders unless they are shown to be definitely polyphyletic or paraphyletic. This has resulted in several well-known families being combined into one or divided into two. Informal names are temporarily being used for certain groups until we have more confidence that we truly understand their phylogeny. An especially important example for us involves angiosperms. Until recently, we believed that all angiosperms were either monocots or dicots, that the monocot-dicot divergence occurred extremely early. However, now there is strong evidence that early angiosperms diverged into several clades before that happened. And the living descendants of those early diverging clades are no longer called dicots. They are just called the basal angiosperms and have not been given a formal taxonomic name. The rest of the dicots are called eudicots, and this too is an informal name. The old word dicot has to be abandoned because the group it referred to is now known to be paraphyletic. Other types of classification systems. Artificial systems of classification. The natural system of classification, which attempts to follow the evolutionary history of the organisms being categorized, is only one of several types of classification system. Another fundamental type is an artificial classification system, in which several key characters, often very easy to observe, are chosen as the basis of classification. Good examples are roadside floras and picture guides to plants, birds, and mammals of national and state parks. The botanical classifications in these are often based primarily on flower color. All plants with white flowers are grouped together, as are all those with red flowers, and so on. Within each category, the next classification category might be the plant's habit. Trees are grouped together, as are shrubs, herbs, vines, and so on. These systems are described as artificial because many plants in a category are not closely related to each other by descent from a common ancestor. Furthermore, they are separated from their close natural relatives in other categories simply because they differ in flower color or habit. Artificial systems typically have the goal of easy plant identification by means of obvious characters such as flower color and plant habit. Alternatively, an artificial system may be designed to group together plants with economically or scientifically important features. From a practical standpoint, carpenters and woodworkers are more interested in color, texture, grain, and hardness of a wood than its phylogeny. Gardeners might classify plants according to their ability to tolerate shade, full sun, frost, or alkaline soils. Plants and people. Box 18-3 Genealogy versus clades. Your family history is the opposite of a clade. It is easy to confuse cladistics with genealogy. When tracing your family tree, you consider your parents, their parents, the next set of parents, and so on into your past. You are trying to locate your past relatives, but the real point of a genealogy is to map out all the people who might have contributed alleles to your own genome. You received all your alleles from your 2 parents, but those alleles came from your 4 grandparents, your 8 great-grandparents, and your 4,096 great-grandparents, and so on. At each generation you are considering the potential input of new alleles, and also you double the number of ancestors you are considering. Your family tree becomes broader as you go farther back. A cladogram is just the opposite. We are interested in one single set of ancestors or founder individuals and how their progeny change with time. The ancestral group is genetically isolated from all other groups, so their only source of new alleles is mutations within the descendants. A cladogram attempts to map out the origins of new alleles or of important new combinations of alleles that lead to detectable changes in phenotype. A genealogy maps the genetic convergence of characters from many ancestors into one descendant over a few generations. A phylogeny maps the evolutionary divergence of characters and progeny over thousands or millions of generations. Artificial classifications are only used as adjuncts to natural systems. As physiologists examine the phylogeny of all species with C metabolism, they find that C metabolism has evolved several times. Therefore, all C metabolisms are not expected to be identical. Similarly, Someone studying the metabolism of petal pigmentation might classify flowers artificially according to color, deciding to investigate the synthesis of red pigment first. But there are two very different types of red pigment, anthocyanins in most angiosperms and betalains in the order Caryophyllales. If some scientists were working with red rose flowers and others were studying red beets, they would get inexplicably conflicting results about the enzymes and pathways involved in pigment synthesis. Figure 18-6, a cladogram that represents the historical view that all angiosperms were either monocots or dicots. These had the formal taxonomic rank of class, Monocotyledone and Dicotyledone in some books, Liliopsida and Magnoliopsida in others. This very simplified cladogram summarizes a current hypothesis of evolutionary relationships within angiosperms. The basal angiosperms diverged from early angiosperms before the monocot-dicot split. All basal angiosperms had been known as dicots, as had all eudicots, but that concept of dicot would be a paraphyletic group, as it would leave out some descendants of the early angiosperms. We do not use paraphyletic clades, and therefore, we need a new name for the basal angiosperms and for the dicots to the right of monocots. Classification systems for fossils. A third type of classification, used for fossil organisms, combines features of both artificial and natural systems. The goal is to understand the evolution of the fossil and to identify both its ancestors and its relatives that might have later evolved into other species. This requires a natural system. Typically, we do not know much about the fossil. Thus, superficially similar ones are grouped together for convenience, a basically artificial system. The groupings are form genera. All fossils with the same basic form or structure are classified together. For example, a piece of fossil wood similar to the wood of modern pines, spruces, and larches is classified in the form genus Ptyioxylon. If the piece of fossil wood was part of a branch with leaves and cones attached, there would probably be enough characters present to allow us to assign the wood to a natural genus, but if the fossil contains only wood, not enough characters are present to determine whether it came from an ancient pine, spruce, larch, or some other group that has since become extinct. Figure 18-7, this Lapanda's orchid is fully grown. Even in full flower, it could be overlooked easily. The photo is twice life-size. Taxonomic studies. People have been hunting for and discovering new plant types since before recorded history to find not only new food plants, but also ornamental plants and beautiful woods. Exploration continues today as biologists enter new areas that either have never been explored or have been visited only once or twice. Tropical rainforests of Brazil, Central America, Africa, and Southeast Asia are so poorly explored that any serious expedition returns with previously unknown species of plants and animals. Some plants are less than 1 centimeter tall when fully grown and have an extremely limited range. It is easy to walk past them without noticing them. On the other hand, some large plants are too big to overlook, but they grow in only one small area and can be missed easily by explorers. Finally, a species may resemble others so closely that a scientist may ignore it, thinking it is already well known. The fact that it is a new species might be revealed only by careful study of certain details. Botanical exploration is also concerned with discovery of new facts about already recognized species. No species is made up of absolutely identical individuals. Therefore, numerous samples must be collected to gather information about the variability of its features as well as its geographical and ecological range. Exploration may be dedicated to gathering either seeds or live material so that the plant can be propagated for research, horticulture, or food. Now that recombinant DNA techniques make it possible to transfer genes from one species to another, live preservation of as many species as possible is particularly important. Preliminary studies of newly collected plants include many diverse activities. Specimens must first be identified using diagnostic keys and personal knowledge. An experienced taxonomist often recognizes the family of an unfamiliar species almost immediately. Even the genus may be obvious. If so, further identification is usually simple. In an unfamiliar region in which many species are unknown, identification can be more difficult, and numerous characters must be studied and compared with those in published descriptions of the plants of the area. If the plant can be identified, it may be studied to see if it provides new information about the species. Even if it turns out to be absolutely typical in every way, the two is important. If the specimen cannot be identified or if it does not match any description, the taxonomist may suspect that it is a new species. Typically, the specimen is then sent to a specialist experienced with the species most closely related to the specimen. Declaring a plant to be a new species is easy. Proving it and describing it properly are difficult. Anyone can declare a plant to be a new species, name it, and write about it, even if it is obviously not a new species. This has caused countless problems. To overcome this, taxonomists from all over the world have established an international code of botanical nomenclature that describes precisely the steps necessary for naming a new species. A valid name, one never previously used, must be declared and must be accompanied by a detailed description of the species in Latin and usually also in English, French, German, Spanish, or Russian. The name and description must be published in a widely circulated journal, a step that prevents many problems. The journal's editors send the description to at least 2 independent specialists to verify that it is a previously unknown species and that the name has never been used before. The description must also include the designation of a type specimen. This is a single preserved plant that truly carries the name. When new species are named, very little is known about them. As more research is carried out in the following years, enough variation may be discovered to warrant the recognition of a second species. For example, the range of leaf sizes, types of trichomes, and types of nectaries may warrant classification into 2 or 3 species. The original type specimen determines which type of leaf size, trichome, and nectary goes with the first name. When doubt arises as to which type of plant goes with which name,